Mary. Really appreciate your talk, and I'm happy to welcome now Robert Green from the Brigham and Women's and the Broad Institute to the stage to give his presentation. Welcome, Robert.
Thank you very much. It's a pleasure and a privilege to be here. It's my first time at this meeting, and I love it already. I like the small room and the common presentations, and all of the friends and colleagues who are here. I'm going to do a little bit different from my usual talk today and try to address the controversy of sequencing healthy individuals with empiric data. And first, I want to just make my disclosures. I do consult for several companies, and they are there. And I also am a co-founder of a telemedicine network for distributed genomics expertise called Genomic. And I guess when you think about this audience and you think about what we've been talking about here and over the last few days, one of the questions is, why is this even controversial? Don't we all believe that individuals are going to be sequenced, that there's going to be a kind of preemptive genomics embedded in our healthcare system? And I would bet that most of us do. But when I've done polls in other speaking situations, people radically differ on the time scale they think this is going to take and how it's actually going to take place. And some people still, some very strong voices are still saying this is never going to happen, it should not happen, it's inappropriate. And they have really interesting and appropriate reasons for considering that, mostly growing out of the public health notion that when you screen for a whole lot of very low probability events, you're going to get way more false positives than you get true positives. So this is a legitimate controversy that is going to dog us for some years. And what we've been focusing on in the Genomes to Research program, Genomes to People Research program, is trying to generate empirical evidence. Now, what are the actual questions that I think are powering this controversy? One is still, is genomic information toxic? Another is, are participants or their providers going to misunderstand and misuse genomic information? And essentially, are the harms and costs going to outweigh the benefits? So our program has launched a number of research studies, which I call, there are a lot of titles, but it's really all about small data. And we focus like a laser beam on the medical, behavioral, and economic outcomes associated with using genomic information, either in medical systems or in society at large in terms of some DTC operations. And today I'm going to talk about a couple of these and try to share with you what I think are some of the most exciting results, including some of the most recent and unpublished results. I think most of us are convinced at this point that genomic information is not toxic, even when it's unexpected, even when it's unanticipated, even when it's bad news and is sometimes around untreatable conditions. And as most of you know, we researched this in terms of the REVEAL study for quite a number of years. And I won't go into this since most of you know that people who elect themselves, who self-select to get even scary information around APOE and Alzheimer's disease typically do not have any kind of distressing information. This has been borne out in large-scale direct-to-consumer genotyping across the nation. And it certainly don't want to be cavalier in the sense that no one is ever upset. But what we've seen both in our highly controlled randomized trials and in the millions of people who've received this information is that there does not appear to be very much distress. And when it is there, it does not appear to be very long-standing. We've also really tried to dig deeply into what people understand about genomic information. And we've really used direct-to-consumer as a model for this in terms of not just one, but multiple responses, whether people are confusing risk variants with deterministic variants, whether they're distressed about one thing or another, whether their family history or personal history seems to shade the way that they understand information, whether that understanding persists over time, how it relates to the way they first understood things. It's really fascinating. It gets deeply psychological in terms of what prejudices and biases they brought to the table when they got their information and how that influences the lens through which they get their genomic information now. We've looked at ethnicity, at socioeconomic status, family history, a whole bunch of things. And so there are some concerns here. There is, just at a high level, there are genuine misunderstandings in consumers. and they are persistent, and those are echoed among their providers. And so this is a concern that, particularly as people start to imagine how they're going to medically react to genomic information. Many have a very clear sense and a good comprehension of that. I don't think it's a national emergency, but there is clearly segments of this population that misunderstand, some misunderstand deeply what's going on. And I think that's a concern that we have to be mindful of. But I want to spend the rest of the talk really on sequencing and talk about the medical, behavioral, and economic outcomes associated not with sequencing for indication. I don't think that's particularly controversial at all at this point, but with unanticipated findings from sequencing. And I like that term unanticipated, which we can use for secondary findings when we're doing an indication-based test, or we can use it for screening people. But I like it. I like to lump it all under unanticipated findings. And the first study I want to talk about is our MedSeq pilot project, in which we brought our randomized trial philosophy to the conduct and to the communication and follow-up of whole genome sequencing. So basically, if information is like a pill, we provided whole genome sequencing, a report, to both the patient and their physician. We did this in people who had a genetic condition and people who were ostensibly healthy, and I'm really going to focus on the ostensibly healthy people. And as you can see, it's just like any other randomized trial. You randomize people into standard of care plus whole genome sequencing. In this case, we also used family history, of rigorous family history, so that there was something for the provider and the patient to react to. And along the way, with the critical leadership of Heidi Rehm in our organization, we helped refine the variant classification that we would use for this and how we would actually classify these variants and put them into a report. And again, in collaboration with partners, Laboratory for Molecular Medicine, we created a single-page report and importantly trained up the primary care doctors on this report so that it was not unfamiliar to them. When they had questions in a non-threatening environment, we explored what these questions were. We had sort of mock reports that they could learn to talk to their patients around. And we found that with a very modest amount of training, just like you don't have to be a radiologist to read a radiology report, you don't have to be a chemist to read a chemistry report, we found that people could, primary care providers, sure, they were self-selected, yes, they were in an academic setting, yes, they donated a certain amount of time to this situation, but they became more and more comfortable with this kind of report and with talking about this kind of report with their patients. We even put in a little bit of pharmacogenomic associations before those were standardized phenotypes in order to sort of do proof of concept. So we'd have sections on monogenic results, carrier risk, pharmacogenomic associations, and blood group serotyping. So let me click you through some of the medical, behavioral, and economic outcomes. We really have a lot of data here, so just some of the high points. One of the great high points of this study is that there's more there than we expected. We're not talking about the ACMG59 genes. We're not talking about the Geisinger 80. We're not talking about the Invitae 139 or the Understand Your Genome 1691. We're talking about looking at close to 5,000 well-associated Mendelian genes. And when you do that, not only does everybody, almost everybody, carry recessive carrier variants, that's not a surprise, but look in this small sample size, but unselected sample size, at the number of people who were carrying what we're calling Mendelian disease risk. And by that we mean a dominant, a pathogenic or likely pathogenic variant for a dominant condition, or biallelic pathogenic or likely pathogenic variants for recessive condition. And in this little cohort, it's 21%. Small sample size, but completely unselected. And I'll show you others that seem to recapitulate that. And then when you go back and you take those individuals in whom you found something and you dig down into the history, the family history, and into some deep phenotyping, you start to find that there are pieces of disease, fragments of disease that are actually present. It's not all about risk. A surprising number of these individuals actually have features of the disease in question. White spots in the fundi, odd rashes, elevated transferrin level. And of course, there are the ones that have nothing, but they continue to be at risk for future diseases. Now, the behavioral outcomes are pretty straightforward. As in so many of our studies and so many of those that others have done, there simply does not seem to be issues for anxiety or depression in people who've learned even about scary cancer, cardiovascular sudden death variants, as long as they elected to pursue this and they got some kind of briefing on what the pros, cons, and risks were. And the economic outcomes are also fascinating. There's been more ink spilled, particularly when I go to England and talk about this in the context of a national health service where they're very focused on costs. There's been more ink spilled about, oh my gosh, if you get this kind of risk information, you're going to blow up medical outcomes and costs. Downstream medical outcomes are going to be ridiculous because everybody's going to go out and they're going to absolutely order all sorts of medical tests, and you're going to break the medical care system. And in point of fact, when you did this in the context of primary care doctors who had an understanding of risk, who could reassure the patient, who could interpret, who could say, we're already doing this, or we've already checked you for this, your EKG last time was normal, or we think, let me think about your family history and we'll talk about it some more. When you had some sort of mitigating gatekeeper like that, who had a minimal amount of training, the costs were almost insignificant in MedSeq in terms of downstream additional findings. Now let me switch to the BabySeq project, which is another project funded by NHGRI. This one technically through the Child Institute, but both of these projects funded through NHGRI and this one through the Child Institute. And the question, of course, is that the notion of sequencing newborn babies has captured everybody's imagination for a long time. This quote from Francis Collins reminds us of that. People have been talking about this for a long time, but very few people have been doing it, at least under a rigorous methodology. And once again, by putting ourselves at equipoise, neither being obstructionistic about it, saying, oh God, we can't do this, nor being cheerleaders for it and saying we must do this, let's do this in some marketing vein or create some commercial entity. We try to put ourselves right down the middle in equipoise and say, let's create a methodology for bringing people into this, get the people who are self-selecting and truly randomize them, because those are the people whose reactions matter. And again, preliminary medical outcomes here are quite remarkable. Out of the 163 newborns sequenced, we have found 11% or 18 cases with, again, this Mendelian risk variance, findings predictive of a monogenic disease. And 89% with no findings or only carrier status findings. Now, looking again more deeply, and we haven't completed this, so there could be more to uncover. Looking more deeply at these 18 cases, we have found 4 cases where there was actual links between the findings that were dug up in the case and the gene variant. And in one of these, the child was recognized as ill, but it was completely not connected with this particular mutation. And in 3 of them, the findings that unveiled fragments of clinical disease were completely undiscovered and unrecognized by the entire medical team that was caring for these children. And then we also found 8 other families where, when we went back with the information, there was a revealed family history. In other words, are you sure you don't know of anything that might be affecting cardiac disease in your family? Well, now that you mention it, Uncle Joe. And so there was additional family history made. So it's further evidence that we can't just go at this from clinical presentations, because clinical presentations are missing things. We can't just go at this from family history. But if we also legitimize the path of DNA first, we can iterate on whatever we find and up that Bayesian prior probability from being extremely low to possibly higher and higher. And if that's our model, instead of thinking about genetic information as a diagnosis or a non-diagnosis, but thinking of it as a Bayesian risk factor that we iterate upon and move up or down on a scale, then I think it's a really different and much more effective model. Again, the BabySeq behavioral outcomes, no difference on randomization arms between vulnerable baby syndrome, mother infant bonding, but as you might expect, in those families in whom a monogenic finding was discovered, there is a sense of an increased vulnerability among the parents of that child. This, by the way, demonstrates that our measures have some sensitivity, but it also, I think, is arguably appropriate. These are families who found that their child has a monogenic risk variant, and they're not devastated by it by any means, but they are aware and they recognize that their child has some additional vulnerability. And the economic outcomes it's a little bit more of a complicated picture. There does seem to be a trend toward increased spending in the babies who, both the sick babies and the well babies who receive sequencing. You can see that there's a trend over the first 3 months and again over the first 6 months in this, much higher, of course, for the sick babies than the well babies. But in this small sample size so far, not statistically significant. Now, this is a mixed bag. I'm not arguing that there's no increase in cost. The question, of course, is whether the increase in cost is going to be worth the value of the information ascertained. But wait, there's more. The problem of penetrance is at the heart of all this. How many of these monogenic risk variants are going to manifest over time? And one of the limitations we've had, I think, for so long is, number one, of course, we've looked at penetrance within families, and we haven't really looked at penetrance on a population basis. And number two, we've really looked at penetrance in relatively short time windows. So what happens when you look at penetrance over a much extended time window? And I'm particularly proud of this paper that we did in conjunction with Seq and his group, because we took individuals from the Framingham Heart Study and the Jackson Heart Study. And these was right after the first group had been exome sequenced, and it was only 462 people in Framingham and 3218 in Jackson. And we said, okay, some of these people have been followed, even in Jackson, for 5 to 10 years, and in Framingham for 20 to 30 years. And we said, what's going to happen if you sort of look across the time period in which they were followed, and you ask how many people with some sort of Mendelian mutations actually developed not just the named condition, not something that appeared in their medical record, but developed some feature of the disease that might not even have reached diagnostic criteria yet. And we could do that for some of the cardiac diseases, particularly the cardiomyopathies, in the Framingham and Jackson studies, because they weren't just keeping track of EHR diagnoses, they were measuring serial septal heart thickness and so forth. And so therefore, we did this. It was a labor of love. We spent 2 years unfunded doing this with volunteer efforts from some of the first authors there to sequence these, to classify these variants, and then to correlate them with the phenotype information that had been collected. And the results, again, small sample size, but statistically significant. That just looking now at that time, the ACMG 56, there were more phenotypes manifesting in the people who carried mutations than in those who did not. Now you might say, well, duh, if people are carrying mutations as a group, I expect them to have more phenotypes. But if you look at the percentages there, they're creeping up. Very small sample size, just a handful, but over an average of 20 years, 4 out of 5 of the people in the Framingham had, and 7 out of 26 in Jackson over a much shorter time period. So it raises the question, at least, a possible proof of concept that raises the question, if we followed these Mendelian risk variants long enough, would we see a much higher level of penetrance, particularly if you take into account that some of these with thickened heart walls, absolutely abnormal thickened heart walls, had not yet had a formal diagnosis of cardiomyopathy. So when we talk about going into the medical records, like that wonderful Vanderbilt paper, and they talk about hidden Mendelian disease, how much more is hidden that never even makes it to the diagnostic level yet in the EHR? And you could argue, if you're a cynic and a nihilist, you could argue, well, do I even care if it hasn't reached the point where it's providing symptoms to those people and pushing them into the medical care system? And I would argue, absolutely, we must care, because this is the dream of precision medicine, to try to find fragments of disease or find the anticipation of disease before it's really getting started, before it causes those symptoms, and prevent it. You know, there's this wonderful term that gets bandied around in the TED Talks called the Umwelt, which is the portion of the visible light spectrum that we can actually see, and reminds you of all of the portion of the light, of the electromagnetic radiation that we cannot see. And I think in genetics for so long, we've been really focusing under the lamppost at the things that we could test for, at the things that, and we've been avoiding the darkness around the lamppost, to mix my light metaphors badly, the darkness around the lamppost in a really important way. And that as we now look more broadly, more deeply, more genes, more careful classification, and then circle back and look deeply at the phenotypes, we're becoming aware of more and more aspects of the disease. I just like this because most people make this point with an iceberg, and I wanted to make it a different way. Okay, so my actual last slide is really the power of, is really extolling the power of small data. Because how many times have you been hearing the power of big data? I just want to fight for the little guy here. The small data power is that by looking at small sample sizes and really digging down deeply, I think we can anticipate the future in ways that the big data collections aren't quite yet able to do. And so I believe that in small sample sizes, we've shown that robust monogenic risk can be identified in 11 to 18% of 3 separate populations. That's MedSeq, BabySeq, and out of the first 60 or so of our pilot study in the United States military, we're the first study to be sequencing healthy active duty military, and we're finding the exact same numbers there. So consistently, we're finding a much higher proportion in the aggregate than I think has been suspected when you look at the entire, if you want to call it that, the Mendeliome. I think we never want to be cavalier about people's reactions. All of us in the clinic have seen terrible distress, and we know we need to be sensitive to it. But I think we're seeing study after study after study where the psychological distress among people who elect to pursue genetic information for themselves, particularly predictive information, who've made that decision, weren't forced into it. It's not like we went out and made them learn something, but they've elected to do it with even just the most basic understanding of the pros and cons. They take that information pretty well, and there simply is vanishingly small amount of distress associated with that. Previously undisclosed and undiscovered medical abnormalities and potentially medical benefits can be identified in multiple domains in approximately a quarter of all those who are being identified with these monogenic risk variants. So I don't know, pick 16% there or even 12%, and you've got a substantial amount of the population that we can discover, if these numbers hold, totally unsuspected fragments of disease due to monogenic conditions. This really puts a funny spin on rare disease, right? I mean, it's no longer rare in the same way. Downstream medical costs are marginally increased, but certainly appear to be lower in scope than were previously expected. These are hard, as other people point out, these are hard studies to do. But by the way, they're much easier in a randomized trial format than they are in all these various modeling formats, because you got people who got it, and you got people who didn't, and you got a controlled situation where you can follow them carefully for the costs that follow. And the penetrance of the Mendeliome may be higher than anticipated over a long time frame, particularly if you realize, I mean, the huge data studies that are pulling from people's electronic health records are limited not only by the distortions that the billing system has on electronic health records, but also by the things which are not yet discovered in people, by the subclinical presentations of cancer, by the subclinical presentations of neurologic disease, the subclinical presentations of heart disease, and so forth. So think about this for a minute also in terms of all the things we didn't talk about in the genome. Think about the recent excitement from Amit and Seq about polygenic risk scores and many others here. Polygenic risk scores that for each condition might have a quintile of the population that approaches the kind of effect size you get with a monogenic. And how many of those do you have to march out from? How many conditions do you have to march out from before you're taking in a large portion of the population who has something? Think about the reproductive implications of recessive carrier states, if that could be made. We've heard about the pharmacogenomic variants, which, it's a very compelling case for a large proportion of medications. And you start to really paint a picture where the negatives are being gradually proven to be smaller than we were worried about. And the positives, I believe we can say, without being hyperbolic, that there's more evidence of potential positive benefit than we initially expected. And so I think we're going to be able to make the case for proactive, population-based genome sequencing in the very near future. And that's good because it's happening. And it's important, though, that we create an unambiguous evidence base that supports it, that we can stand behind, and not that it's happening for commercial reasons, marketing reasons, or as part of the very, very powerful zeitgeist that's propelling this biotechnology along, enthusiasm, technophilia, money, and so forth. So it's really important to acknowledge the leadership team and all of the people who worked, including many people in this room, on the MedSeq Project, with whom we still have the pleasure of working together as we continue to analyze these results, and also the BabySeq Project team. I want to make sure I mentioned that I co-lead this project with Alan Beggs of Boston Children's Hospital. And we're continuing with both of these. We think it's very important to follow the long-term benefits or harms that might accrue in both these populations. We think we've got a lens to follow them that has a higher resolution than most. And so the next time you hear the power of big data, I hope a little voice in your mind will remind you of the power of small data. Thank you very much. Yes, Bob.
Great talk, Robert. Fantastic work. For those of us who are interested in sort of following in your footsteps and trying to implement a preemptive sequencing and don't have 3 MD postdocs to go dig into the EHR deeply and things, have you gotten through experience a sense of where are the efforts best spent? Is it through deep family history? Looking through the problem list or diagnosis list?
It's really everything, Bob. I think it's a tough one. I mean, when I tell colleagues that NHGRI funded the MedSeq project for $10 million and we ended up sequencing 100 people, they're somewhat aghast. But I hope you will believe after this that we used the money for more than sequencing, and part of it was digging down deeply into this. And it's very time intensive. And I think this, however, provides a set of goals for all of this data stream we heard about. And last night we heard about the data stream that Carlos was talking about, from wearables, from new biotech measures, from biological monitoring devices. If you can wear something on your chest that in a Star Trek manner measures your cardiac septal wall growth over time, maybe that's an early warning sign that's way beyond that. But maybe you only buy that if you've got a cardiac risk variant for cardiac hypertrophic myopathy, cardiomyopathy, yeah. Hypertrophic myopathy. So I don't have an answer for a research venue. I'm very grateful to NIH and NHGRI for supporting this, but I hope it lends credence to finding less expensive, less labor-intensive ways of tracking this information in the future.
Robert, I wonder if there's a way to sort of extrapolate forward from where we were 10 years ago to the future with your populations to sort of make an argument for clinical benefit moving forward? Because I think when we often think about clinical benefit, especially in newborns, we think more about kids that are, well, we'll hear about Stephen's presentation after the coffee break, but who sort of have this tangible need to be understood at a genomic level, whereas you've kind of taken the opposite approach. But I think at the end of the day, we don't just want to sequence those kids that have failure to thrive and other problems. We want to sort of sequence every kid so that we can do the kind of future forward evaluation, the sort of penetrance-based evaluations that you're talking about, and maybe even the Star Trek devices, which I'm holding out for personally. But can you sort of do that thought experiment to make an argument for long-term clinical benefit, even from apparently healthy individuals?
Yeah, it's a great question, and I don't know if I can do it well, but I think that it's really true. I think this is here with us. We're going to have to continue to validate this kind of implementation at larger and larger scale. And then before we can even take a breath, I think what's going to be on top of us is proteomics, metabolomics, microbiomics, and then how these work together. And so I think there's going to be, essentially, it's going to be like being at the beach and being hit by a wave. We'll just catch our breath from this wave, and then the next wave is going to come in, and the next wave is going to come in. But it's just like it's important to keep your footing and stand up to those waves. I think it's really important for us to be vigilant about evidence collection, because part of what's driving this, we had a wonderful discussion about this yesterday, but part of what's driving this is enthusiasm for new technology, a desire to do good, a desire to help people, a desire to differentiate your own healthcare system from others, and let's face it, quite a lot of investment money in the society at large. So in order for this to go forward in concert with science, we have to be vigilant, keep our balance. I define that as a kind of equipoise at the beach. And as wave after wave crashes over us, make sure we're standing up to it with the equipoise of evidence generation.
Nice. Okay,
Thank you.
Thanks very much. Let's thank our speakers for the first half of the session. So we're breaking now for coffee. The coffee break areas are next door in the Grand Ballroom D and E and in the Scripps Ballroom. And please come back here around about 11. We'll start right at 11 o'clock for the second half of the session. Thanks very much.